जनों को सादर प्रणाम, नमस्ते। वारणा नगरी में आप सबका हार्दिक स्वागत है। देश के कोने कोने से पधारे और ज्वाइन इस आभासी माध्यम से संचालित एक दिवसीय राष्ट्रीय वेबिनार में श्री वारणा विभाग शिक्षण मंडल संचालित यशवंतराव चव्हाण वारणा महाविद्यालय में आईपीएसी और वनस्पति शास्त्र विभाग की ओर से आयोजित इस संगोष्ठी में आप सभी महानुभावों का मैं प्रभारी प्राचार्य प्रोफेसर डॉक्टर प्रकाश चिकुर्डेकर आप सबका हार्दिक स्वागत करता हूं। मित्रों, आज का दिन एक खास दिन है। विश्व प्रकृति संरक्षण दिन है, अर्थात वर्ल्ड नेचर कंजर्वेशन डे है। इस दिवस के उपलक्ष्य में और सुवर्ण जैसे कि कहा जाए कि ये सुविधा अत्यंत उपयुक्त है और दिन भी अत्यंत प्रासंगिक है। ऐसे मौके पर एक अलग विषय को लेकर रोल ऑफ बायोटेक्नोलॉजी इन बायोडाइवर्सिटी कंजर्वेशन अर्थात जैव विविधता संरक्षण में जैव प्रौद्योगिकी की भूमिका, इस एक अलग विषय पर मित्रों संगोष्ठी का आयोजन हो रहा है। काल के अनुसार हमारी विज्ञान की शाखाएं बदल रही है, उनकी उपशाखाएं भी आज अपना एक अलग स्थान जमा रही है। मेरे अपने महाविद्यालय में इंडस्ट्रियल माइक्रोबायोलॉजी, बायोकेमिस्ट्री जैसे विषय हमने पिछले 5 सालों में शुरू किए हैं। इसका कारण ही यही है कि काल के अनुसार इन विषयों की महत्ता बढ़ रही है। मित्रों, जनसंख्या बढ़ रही है। विज्ञान, तंत्रज्ञान, सूचना प्रौद्योगिकी के क्षेत्र में मनुष्य ने तरक्की की है। आज जो जैव विविधता और उसके संरक्षण की बात कही है और उसे आगे जाकर जैव प्रौद्योगिकी जो है जिसे बायोडाइवर्सिटी कंजर्वेशन कहा जाता है, तो ये कोई हमारे लिए नया विषय नहीं है। मित्रों, मैं तो हिंदी का छात्र हूं, अध्यापक हूं, लेकिन आप तो बहुत गाढ़े पंडित है, विद्वान हैं। आप जानते हैं कि इस विषय की महत्ता आप मुझसे ज्यादा बेहतर जानते हैं। लेकिन जहां तक मेरा ज्ञान है मित्रों कि जैव प्रौद्योगिकी की शुरुआत ईस्वी सन पूर्व 6000 साल पहले से हुई है और जैसे कि ईस्ट जिसे कहा जाता है, ब्रेड बटर में उसका प्रयोग किया गया है उस साल में, तब से लेकर इस विषय को लेकर चर्चा होती रही है, उसकी जरूरतें महसूस होती रही है। मुझे पता है कि 8वीं और 9वीं शताब्दी में, मतलब 1980-90 के दशक में ये बायोडाइवर्सिटी और साथ साथ बायोटेक्नोलॉजी विषय को लेकर काफी चर्चाएं हुई और आज इस मकाम पर, मोड़ पर हम पहुंच चुके हैं कि सारी दुनिया इस क्षेत्र से जुड़ रही है। अर्थात मेरा थोड़ा ज्ञान इस विषय को लेकर कम हो सकता है, लेकिन आज का कोरोना हो या उससे जुड़े अनेक विषय हो, ये इसी के नतीजे हैं। अतः मैं ये इस संगोष्ठी के दरमियान, वेबिनार के दरमियान आप सभी विद्वत जन और महानुभावों को प्रणाम कर, अभिवादन कर ये आह्वान करूंगा कि इस क्षेत्र ने जो तरक्की की है, इस क्षेत्र की तरक्की का उपयोग पूरे दुनिया में बसे मानव जाति, मानव कल्याण की हित के लिए होना चाहिए। जैसे कि आप जानते हैं कि बीटी कपास का बियाना है या कोई बैंगन का बीज है, ये सारे मतलब मनुष्य की जरूरतों को पूरा करने का काम ये बायोडाइवर्सिटी और बायोटेक्नोलॉजी के माध्यम से हो सकती है। अब जैसे कि देख लीजिए कि मनुष्य का अब खेती में काम करने का उनका तरीका है, अब मजदूर नहीं मिल रहे हैं, तो ये जो रोबोटिक टेक्नोलॉजी का प्रयोग किया जा रहा है, जैसे कि गन्ने को काटना है समझ लीजिए, तो समझ लीजिए कि 1 एकड़ खेती में 20 लोग चाहिए, तो अब ये बायोटेक्नोलॉजी का प्रयोग कर उसके जीन्स निकालकर मुझे लगता है कि किसान का जीवन आसान हो। अर्थात सिर्फ गन्ना काटना वाले 20 लोग ये बायोडाइवर्सिटी और ये बायोटेक्नोलॉजी के माध्यम से आप बनाएंगे। यहाँ तक कि ऐसा भी मुझे पता चला कि आजकल ऐसे एक आदमी की आदमी को बनाया जा रहा है जीन्स निकाल निकाल कर, जैसे कि लता मंगेशकर जी की आवाज हो, अमिताभ बच्चन जैसी हाइट हो, माधुरी दीक्षित जैसा चेहरा हो, सलमान खान जैसी बॉडी हो, हर एक का जीन्स निकाल निकाल कर एक अलग प्रकार के किस्म के व्यक्ति को पैदा किया जा रहा है। और जहां तक मेरा ज्ञान है कि अमेरिका में 2 महिला साइंटिस्ट ने इस प्रकार के शोध को बनाया है और 2 व्यक्तियों को, 2 लड़कियों को पैदा किया है। लेकिन यहाँ मनुष्य की जो सोच है, मनुष्य को जो तरक्की के रास्ते है और उन्नति के पथ पर वो पहुंचना चाहता है, विकास की चरम सीमा तक पहुंचना चाहता है, लेकिन उसके 2 पक्ष आप है जानते हैं कि एक सकारात्मक और नकारात्मक। मैं इस संगोष्ठी के बहाने आप सभी लोगों को, महानुभावों को, विद्वज्जनों को आह्वान करूंगा कि इस संगोष्ठी के माध्यम से आप मनुष्य जाति का, सृष्टि का, प्रकृति का कल्याण करने के लिए सोचे, चर्चा करें, विमर्श करें, इस प्रकार का आह्वान करता हूं। और इसी बहाने ये जो बायोटेक्नोलॉजी विषय है या जैव विविधता का है, जैव प्रौद्योगिकी है, तो उसका नकारात्मक पक्ष जो है उससे दूर रहे, नहीं तो राक्षस जैसी स्थिति ये मनुष्य की बन जाएगी। और फिर जो हम मनुष्य के कल्याण के लिए, मनुष्य के हित के लिए, मनुष्य की तरक्की के लिए जो हम शोध लगा रहे हैं, अनुसंधान कर रहे हैं, इससे कोई अर्थ नहीं रहेगा। और 2-4 मतलब गिने चुने लोगों के जो एम्बिशन है, जो उनकी हुकूमत है, उसके बदले सारा संसार, सारी दुनिया खतरे में आ सकती है। इसलिए इस बहाने में आप सभी को नम्र आह्वान करूंगा कि ये जैव विविधता को बचाना हमारा कर्तव्य है और अगली पीढ़ी के लिए बचा कर रखना ये तो हमारी जिम्मेदारी है ही, नहीं तो भविष्य की नई पीढ़ियां हमें कभी माफ नहीं करेंगी। लेकिन साथ साथ उसी बहाने जो नए शोध आएंगे, नए रास्ते निकालेंगे, जैसे कि आप जानते हैं कि मनुष्य का जीवन अब इतना खतरेला होता जा रहा है, कोरोना महामारी का कालखंड है, ऐसे हालात में हम इस प्रकार की आयोजन करें वेबिनार का। जैसे कि ब्लड प्रेशर है, मधुमेह है, जिसे शुगर जानते हैं, तो शुगर के जरिए इंश्योरेंस में भी मुझे लगता है कि ये बायोडाइवर्सिटी और ये बायोटेक्नोलॉजी का प्रयोग कर मनुष्य के जीवन में जो एक बीमारी जड़ जाती है, उसे जड़ से खत्म करने का काम भी आप इस संगोष्ठी के माध्यम से करें। मुझे लगता है कि देश के कोने कोने से बहुत सारे लोग जुड़ रहे हैं, अभी जुड़ने वाले हैं। आंध्र, केरल, कर्नाटक, पंजाब, हरियाणा, मध्य प्रदेश, हैदराबाद, साथ साथ पश्चिम बंगाल से, दिल्ली से लोग जुड़ रहे हैं। मैं उन सब का पहले फिर एक बार तहे दिल से हार्दिक स्वागत करता हूं। आज की इस संगोष्ठी के लिए हमारे संस्था के अध्यक्ष आदरणीय विनय कोरे सावकर साहब जी ने शुभकामनाएं भेजी है। प्रशासकीय अधिकारी डॉक्टर वासंती रासम जी ने हमें शुभकामनाएं दी है। और आज के प्रमुख अतिथि रिसोर्स पर्सन आदरणीय डॉक्टर जय चौहान सर है, जो इस विषय के माहिर है, संशोधक है। और इसी बहाने सभी जुड़े सभी लोगों को मैं फिर एक बार तहे दिल से हार्दिक स्वागत करता हूं। संगोष्ठी के लिए, वेबिनार के लिए शुभकामनाएं प्रदान करता हूं और आशा व्यक्त करता हूं कि एक दिवसीय इस समारोह में कुछ नया मिले, नया अनुसंधान हो और मेरे महाविद्यालय के साथ साथ आप हमेशा जुड़ते रहे, हमें भी कोई पथ प्रदर्शन करे। इसी मंगल कामनाओं के साथ मैं आपसे विदा लेता हूं। धन्यवाद।
Thank you, sir. You have given very brief introduction, the advantages of biotechnology and the possible disadvantages also. And you have instructed all the researchers to think on the negative side and try to keep your research away from such negative impacts. Thank you very much. Now I request Dr. Tarannum Mulla madam for the introduction of today's resource person.
Good morning and welcome to all. Dr. Tarannum is here. It gives me great pleasure to introduce today's resource person, Dr. Jai Kumar Chavan, who have leadership skills with wide range of experience in academics, teaching and research. Dr. Chavan has completed his graduation, post-graduation and research degree from Shivaji University, Kolhapur. He is presently working as Head of Biotechnology Department, Co-Coordinator of Internal Quality Assurance Cell, as a Chairperson, Board of Studies in Biotechnology, Biochemistry and Forensic Science at Yashwantrao Chavan Institute of Science, Satara. As a resource person, he has delivered more than 35 scientific talks at various seminars and workshops. Dr. Chavan has published more than 46 research publications, including book and book chapters. He has completed successfully 5 major research projects supported by various funding agencies like DST, DBT, and UGC. He is working as editor and peer reviewer for 10 international journals, including United States, America, and London, and associated with international collaboration with various institutes, laboratories, and universities. He is life member of Professor H.S. Srivastava Foundation and Society of Biochemistry and Biotechnology. He is currently working as a member board of study at various colleges. Recently, he has been appointed as a research advisor at Nanyang University Academy of Science, Singapore, and he also elected as a young associate Maharashtra Academy of Science. It is indeed a great privilege to have Dr. Chavan with us. Thank you, sir, for accepting our invitation. Now I request Dr. Chavan, sir, please continue the session. Please, sir. Thank you.
Thank you, madam.
Yes.
Chavan, sir.
Thank you, Dr. Tarun,
Yeah.
for kind words. I felt very elated by your words.
Thank you, sir.
Very good morning to all the virtual gathering. Respected chief guest of this event and very active and eminent personality in the field of administration, Professor Vasanti Ram.
Sir, please unmute yourself.
Okay.
Yes, sir.
Chief guest of this event, Professor Vasanti Rasa, Principal of Host Institute, Dr. Prakash Chikurdekar, sir. My teacher during post-graduation at Shivaji University, Kolhapur, Chief Organizer of this event, Captain Dr. S.S. Khote, sir. My colleagues of botany, Dr. S.Y. Jadhav, sir, and Dr. Tarun Mullah, and all the virtual gathering. First of all, I express my wishes on as world is celebrated today. World is celebrating World Nature Conservation Day, as every year it is celebrated on the 28th of the July. And the major reason behind the celebration of this World Nature Conservation Day is to raise the awareness among the every entity of the this society towards the conservation of the plants, either anyways, as most of us knows that there is no need to break your what we can say that your FDs or don't need to invest much of the money for the conservation of the species or the animal species. Just you should have your positive thoughts even in your mind, and definitely it will help to conserve the biodiversity in many ways. So, dear friends, first of all, I thank authorities of the Yashwantrao Chavan Warana Mahavidyalaya Warana for inviting me to talk on the role of biotechnology in the conservation of the biodiversity. As this is the conservation of biodiversity, we are listening since long back, but how the biotechnological implications or the biotechnological involvement helps to conserve the biodiversity, especially the plant biodiversity. That part we are going to deliver. I'm going to deliver in this national webinar. So just a minute, I'm going to share my screen. Oh, sir, is it visible?
Yes, sir. It is loading. Just wait. Yes, sir.
Okay, sir.
Okay, it is in slideshow mode.
Yes, sir.
Okay. Okay, dear friends, as I told that biodiversity is one of the important part.
Sir, it is
and
not in presentation mode.
It's not in presentation mode.
Yeah, please.
Okay, again I will. Sir, is it in presentation mode? Good, sir.
Just wait, sir.
At my end, it looks in the slideshow mode. And again.
Yeah, it is loading, sir. Loading.
Loading.
Yes, presentation is on, sir. Just start the slideshow.
Just confirm the slideshow, sir. I have right now I have put it in on the slideshow mode.
Please turn on full screen, sir. F5, control F5.
I have given the full screen mode.
Okay, no problem, sir. Your slide is visible. Please continue.
Visible?
Yes,
Okay.
sir.
So, dear friends, today we are going to deliver or we are going to discuss about the various aspects of the biodiversity. I'm not going into details about the biodiversity as most of the peoples they are aware about what is the biodiversity, what is the role of biodiversity. I will just highlight the things which are necessary to be there or to be presented before you. So, biodiversity that is nothing but the variety among the life forms on this earth format. But there are different levels of the biodiversity which we are considering in our day-to-day life. That is, first of all, the genetic diversity, then species diversity, then ecosystem diversity. These are the three major levels of the biodiversity which are we are considering during the diversity studies. However, the genetic diversity, so this slide is visible, genetic diversity, species diversity and levels. Good, sir. Okay. These biodiversity levels we are always studying or we are always considering while studying the biodiversities, that is genetic, species as well as the ecosystem diversity. I am not going into detail about the biodiversity. However, as Dr. Sudhakar Khote sir have mentioned that the most of the participants they have participated from various corners of the country. So I should show some of the biodiversity regions or the geographical regions which are specially considered for the biodiversity studies in the Western Ghats, especially in the northern Western Ghats. So, dear friends, where I resided in the northern Western Ghats, here we have one World Heritage Site which was declared by the UNESCO in 2012 as the what we can say that is the karst plateau, which is considered as the world's heritage site. So, dear friends, these karst plateaus are nothing but the top ends or the flat ends of the hills, and this northern Western Ghats is very popular for having such kinds of plateaus, and such kind of plateaus have the very unique plant diversity at their ends. So I am just highlighting the karst plateau where we are observing the several numbers of the various kinds of the vegetation pattern on this karst plateau. This karst plateau is extended within the 25 kilometers, 25 square kilometers, but this much small area which harbors about 411 different types of flowering plant species at a single place within the 25 square kilometer area, and most of them are the endemics. About 24% of the species which are occurred at the karst plateau, those are endemic, and they are not found at all at the other places across the globe. So such kind of biodiversity is occurring at any geographical areas, but I am just introducing a karst plateau, which is considered as the one of the important geographical area to study the biodiversity. There are different types of plants or the flowering plants which are occurring at this end, but the one of the important and very striking feature of this World Heritage Site is the seasonal changes across the plant diversity during the various seasons. If any visitors or any researchers visit this place in the month of May, actually in the season of summer, at that time we will not able to get a single herbaceous flora at this end. At that place only we can see where lateritic plateaus are there on the hilltops. However, once the spring season or once the rainy season starts, at that time we will able to get or we will able to see some flowering plants. Only those are in the small streams which are there on the plateaus. If we visit after every 15 days or the 20 days intervals on this World Heritage Site, at that time we will able to see the very different vegetation patterns on this plateau. So dear friends, once we visit, what we can say that once we visit this karst plateau before the monsoon, that plateau is only having the tree species, but no herbaceous or climbers and other plants are there on this plateau. However, once spring season starts, we are able to see the what we can say the flowering plants in the streams which are there on the hilltops. So once after 20 days, if we visit this karst plateau, at that time we will able to see the glory of utricularia in the full bloom on this karst plateau, along with the mixed vegetation of the ureocolons, what we call it as the gainful. So after the 20 days interval, if we visit the karst plateau, then you will be able to find the full bloomings of the impatiens larvae or impatiens larvae, which we can call it as. Whole plateau is full of the impatiens and what we can call it as at that time. Then there are 5 to 6 different species are observed on this karst plateau. However, impatiens dalgeli is one of the endemic plant species to the karst plateau and nearby areas, including the Mahabaleshwar in the northern Western Maharashtra. Dr. Khod sir, the slides are visible or
Yes sir, visible sir.
Okay, impatiens slide दिसते म्हणजे मला ते पुढे एक्सप्लेन करताना बरं पडतं. Okay.
Yes sir.
Okay, sir. Once this, after this, if we visit the karst plateau at the end of this season, rainy season, at that time, the visitors or the researchers or those are those who visits this Karst Plateau, they are able to find, or they are able to see the different types of the Mickey Mouse species, which is scientifically which is called as this Mythia. There are around 7 different species which are reported from the very small area from the Karst Plateau. These are the some of the glories of this Karst Plateau, or the diversity of the plant species which are occurring at a particular space. One more striking feature of this biodiversity is, I can say, the hotspot, because over 24% plants are endemic to this region. So this is the Strobilanthus, which is popularly called as the Toplicarby. And one of the striking feature of this plant is this plant gets flowered once after the 7 to 9 years. So such kind of diversity of the plants which is occurring at the Karst Plateau, which is considered as the one of the world's heritage site. So dear friends, biodiversity is nothing but, as I told, the diversity among the life forms of the plants as well as the animals. But if we consider the plant diversity, there are different types of the plants in the nature. As I told initially, there are some medicinal plants are there, some plants which are useful as a wilder relatives in the breeding programs in the agricultural practices. Then some of the plants which are endemic. Then some of the plants which are comes under the IUCN categories of the threatened plants, either rare, endangered, critical endangered, or extinct, or the vulnerable. All these categories plants, and this group of the plants which considers which comes under the biodiversity. I am taking some of the examples of this from this biodiversity. First thing is the wild relatives of the crop plants. These are gaining much interest in the agricultural field because just now Principal Dr. Chikordekar sir mentioned that the gene transfer technology is there, and some of the elite clones we are producing using the biotechnology. Similarly, we are using the wild relatives of the crop plants in various ways to improve our existing agricultural crops. This is the example, that is the Vigna khandalensis. How we can say that how diversity is there? If we consider the genus Vigna, which mainly comprises our Vigna mungo, then Vigna radiata, and other our species we are using in our day-to-day diets. However, if we see the diversity among the habit of this plant, whole Vigna genus is the creepers. They are growing along with the soil. However, this is the only species in the genus Vigna which is erect species, which grows up to 5 to 6 feet. So this is the only species in the genus Vigna which is different, or which is diverse from the other species of the Vigna. And most importantly, nowadays people are taking concern of this species because the seeds of this species are very rich in the protein source, and nowadays people are domesticating this plant species for the future perspectives. So dear friends, as I told, medicinal plants are also comprising the major source of the diversity or the plant diversity on this earth. Many of the plant species, or the many of the medicinal plant species, they are found in various corners of the globe. But nowadays, due to the illegal collection of this medicinal plants, the diversity of the medicinal plants, or the occurrence of the medicinal plants on this earth's planet, you get certain barriers, or certain, what we can call it as they are decreasing day by day due to the illegal and indiscriminate collection. So how biotechnology can survive these plants, or will be useful to conserve these plants, I will get into in the next slides. However, nowadays this is the case, that is Saptarangi, which is scientifically called as the Salacia chinensis. That particular plant is used for the treating of the cancers. Mangiferin drug is there in the roots of this plant species, and roots are illegally collected from the various corners of the northern Western Ghats, especially from the Amboli region in the northern Western Ghats. So most of the times the police persons, they are taking the actions, but even though this illegal collection practice is increasing day by day. So for conservation or the domestication practices, how this biotechnology can help, that we will see in the next few parts. So dear friends, nowadays, as I told, most of the plant species from this Earth planet, they are on the verge of extinction only because of two major threats. One is the natural, which is very less. Natural causes of the threats to the biodiversity are very rare. However, on contrary, human-made or the human beings are the major threat to the plant diversity on this Earth planet. We are considering these human-made activities under the anthropogenic causes, where human beings are involved to reduce or to threat the biological diversity of the plants as well as the animals on this planet. How we are going to threat or becoming the barrier to survive the various plants on this Earth planet, we are going to modify the habitats of the natural plants as well as the animals in the several ways. Then we are over exploiting the number of medicinal plants from the natural origins. Then innovation by the exotic plant species from the other countries. Then pollution is there. Then due to the pollution, what we can call it as the aquatic flora and fauna is drastically affecting their survival in the current situation. Nowadays, for fulfilling the food and other requirements of the human beings, more and more land is coming under the agriculture. So these are some of the threats which are considered to be the for the what we can say that during the discussions on the biological diversity conservation or the biodiversity conservation. Dear friends, though we are aware that which plant gives which benefits to the human beings, or why the plants are becoming the extinct from the natural sources. So considering this, we should what we can say that we should thought about why should we conserve the biodiversity. What is the necessity of conservation of biodiversity? In several ways, the biodiversity gives us, so biodiversity provides several things to healthy and stable lifestyle of the human beings. So in this concern, we should conserve the biodiversity not only for ourselves, but it should be for the future sustenance or the future generations. So dear friends, if we think we have to conserve the biodiversity, so what are the ways or what are the routes which are available in front of us for conservation of this plant species? Either we can conserve through the ex situ conservation ways or in situ conservation ways. As ex situ conservation means that is the outside their natural habitats. There are certain examples. If you are conserving the animals, you can take that animals or get that animals from their natural localities, and we can conserve it in the various kinds of zoos. Then what we can say that if we have to conserve the plant species, we can collect it from the natural sources. When we are going to collect it from natural sources, when those natural plants are facing some problems, then and then only we can take such kinds of plants into the other habitats like the seed banks, then botanical gardens, then your natural reserves, natural parks, marine parks, biodiversity spheres or the biospheres. Those are considered as some of the in situ conservation techniques, which means conservation of that plants or the animals in their natural origin. So, dear friends, when we are going to use the biotechnological tools for conservation of the plants, especially now we will focus on the plant diversity or the plants conservation. There are several routes to conserve the plants. Most popular route, as you people knows, that is the our conventional vegetative propagation techniques. There are several techniques, right from seed germination to stem cuttings, layering. There are different techniques which we can use to propagate the plants. But when we can use the biotechnology, when several other routes doesn't work for a particular species, then we can use the biotechnological tools to conserve such a plant species. So which are the, apart from biotechnology, what are the probable other ways to conserve the biotechnology? First, we can consider it as habitat protection. As I told, we should not disturb the what we can exact habitat where this plant currently situated or the animals currently situated. We should not disturb their existing habitats. Then one can use the establishment of the field gene bank. Then development of package of practices, which means that we can develop a package of practices for propagation of that plant species by various vegetative propagation techniques, various irrigation, various nutritional requirements. So we need to develop a particular protocol for conservation of that particular species. Then coordinated efforts by the various funding agencies, research groups, forest officials. It is not a duty of a single person to conserve the plant species. If coordinated efforts are there, if funding, especially most of the Indian funding agencies like University Grant Commission, right from then DBT is there, DST is there, MOEF is there. They are providing the funds to conserve the plants. But the involvement of researchers and the forest officials is the must, as well as the peoples. Because what will happen if funding agency provides the funds, researchers will develop the various propagation techniques, but forest officials and our other local peoples, which their involvement is very much necessary to monitor the whatever plantlets we are transferring into the nature by using the various techniques. Researcher can replant, but whatever the regrowth as well as the development of the plant in the nature, such kind of recordings or the monitorings are necessary. So coordinated efforts will be beneficial to the conservation of the any rare, endangered and medicinally important plants. So study of population genetics is also one of the important and viable route for the conservation of the plants. Then training forest officials. If we train the forest officials about the which are the plants in their particular area, they are under which category, which plants are less in numbers in their particular area, which is under endemic category, which is under endangered category, which is medicinal plants. So if we train the forest officials, they will take care of the such kinds of plants in their particular area. Then one more way is the technology development for maintenance and storage of the tubers. Then study of pollination biology. This is the most important area where the researchers can work, because most of the plant species are on the verge of extinction due to the reproductive barriers. There is a very scarcity of what we can say the pollinators. So there are pollination barriers. That's why most of the plant species, they are not producing the flowers and the seeds. So that's why they are becoming the rare. If one can study the what are the exact pollination barriers in such kinds of species, so at that time we will get the what is the exact reason behind that limitations of the flowering, limitations of particular plant species. Then restoration programs, which means that we can reintroduce the particular plant species by propagating at a particular place, and again it is replenished or it is again reintroduced into the wild or the natural populations. These are the other ways of the conservation of the biodiversity or the plant diversity. But when all these routes doesn't work for a particular species, then we should go for the biotechnology. So dear friends, which are the areas which we can inculcate that areas in the biodiversity conservation? We can consider the different areas of the biotechnology depending on their color patterns. We are using these symbols. If we say red biotechnology, which is associated with the health, medicinal diagnosis. If we consider the green biotechnology, which is associated with the agriculture and environmental issues or the improvement of the agricultural traits in the plants using the biodiversity. Then several areas, they're like blue biotechnology, yellow biotechnology, brown biotechnology. Nowadays, it is a hot topic of gold biotechnology, which is especially associated with the nanobiotechnology. As day by day, the nanobiotechnology, various kinds of the nanoparticles, nanotubes, nanowires, all these things which we are using in the various ways as an antimicrobial agent, as an drug delivery agent. So this area is also one of the hot topic nowadays. But on contrary, dear friends, there is one branch of biotechnology, that is the dark biotechnology. As Principal Chikodekar sir rightly mentioned that as like the advantages of biotechnology, few of the disadvantages of biotechnology are also there. So nowadays we are facing across the globe, that is the COVID-19 crisis or the pandemic. And this is the outcome of the biotechnology or the dark biotechnology, dear friends. As most of the people is considering that COVID-19, which is a virulent strain of that particular virus, which is developed in the I am not going to take the name of that particular country, some of the country, and they have transferred from one region to another region, and it is a so this is the such kinds of the dark biotechnology or the disadvantage of the biotechnology. It should not be there, because if such conditions remain same in the future, then there will not be the need of guns and other things, other equipments for the fights or the wars between some of the countries. There will be a bio war or the bioterrorism in the future. So such kinds of the dark sides of the biotechnology should be hide. It should not be taken the advantages of any country in this particular aspects. So dear friends, which biotechnological tools we can use for the conservation of the biodiversity? First and most important tool of the biotechnology, which is used in the conservation of plants, either medicinal or either rare, endangered and threatened. The plant tissue culture is the one route which is commonly used to conserve the plant diversity. Then molecular marker technology. There are different types of molecular markers are there. That markers are also involved in the identification of the genetic diversity and designing the policy against the conservation of that particular plant species. Then plant DNA bank is there, genetic manipulation is there, several genetic transformation techniques are there. We are morely focusing on the plant tissue culture, and then rest of the we will highlight the other biotechnological tools which are implied in the conservation of the biotechnology. So dear friends, this is the plant tissue culture technique. As I told, plant tissue culture technique, few of the students, they are joined for this webinar. So I will go through some fundamentals of the plant tissue culture, and then I will highlight the what are the advancements in the plant tissue culture nowadays for the conservation or the propagation of the plants. So in the plant tissue culture, we are using various techniques like micropropagation, somatic embryogenesis, protoplast culture. bulbulate, rhizome, tuber formation, in vitro gene bank, somacronal variations. These are some of the side branches of the plant tissue culture which we are using for the conservation of biodiversity. So dear friends, plant tissue culture is nothing but the cultivation of the plant seeds or the either plant parts under controlled conditions. What controlled conditions are there, or what are the exact difference between the cultivation of plants in the in vitro conditions, that is controlled conditions, and on contrary we are growing the plants on uncontrolled conditions, uncontrolled or the field conditions, where we cannot control the what we can say the nutritional requirements, then temperature requirement, light requirement. So on contrary, we can control all these things in a particular space, that is in the plant tissue culture laboratory, where we can control all these things. We can control the microbial contamination, we can control the temperature requirement, light requirement, okay, humidity requirement. So why plant tissue culture technique should be preferred in the plant tissue culture, in the biodiversity conservation? Because, as I told, rest of the roots of the conservation fails, then only we are going for the plant tissue culture. So what is the advantage of plant tissue culture over the conventional propagation technique? That is, for the conventional propagation techniques, what we need is the seed, we need the stem cuttings, or in the few of the plants we need the leaf cuttings. But in some of the plant species, such kinds of the explant doesn't work. So for that purpose, we can, or one can use the plant tissue culture technique for the propagation of the plants, because any of the plant part of a particular species can be used as the source of explants for production of the new plant species, right from root, stem, leaf, flower bud, axis, anthers, embryo, every part of the plant we can use as the source of explants, and that from that explants we can provide, or we can develop number of plantlets within a stipulated time. So that's why plant tissue culture is one of the important biotechnological tool which is involved in the nowadays across the globe. Through the plant tissue culture technique, several plants are conserved. We are going to see in the latter part of this presentation success stories of the plant tissue culture technique derived conservation routes. So this technique, as I told, this technique is dependent on the two important characteristic features of the cell. That is, the first is the totipotency. That is, one can know that the whole plant can be developed from the single cell, that is the totipotency. And one more feature of the cell, plant cell, that is the plasticity. Plasticity, which means the adaptability or the adaptability of the plant cell tissues to the changing environments. As we are taking the explants from the outdoor grown plants, and then we are transferring this plant tissue to the controlled conditions. So at that time, that plant's tissues or the organs should be adaptable or the resist to such conditions. So these two important characteristics are there with the plant cell. So this plant tissue culture technique, which is based on these two characteristics. For culturing of the plant tissue under the controlled conditions, one can require the various chemical compositions or the plant tissue culture media. There are different types of plant tissue culture media, right from the White's medium, which is considered as one of the earliest media for the plant tissue culture. Thereafter, Maharshi again, Cook's media, Maharshi again, Lismer, Niche and Niche, Gamberg V5 medium. And nowadays, in the market, plant-specific plant tissue culture media is there. For example, Knudsen media, which is used for the orchid propagation. Then your banana tissue culture media is different. Gerbera tissue culture media is different. So nowadays, ready-made plant tissue culture media is different, is formulated for various plants. But basic requirement of these plant tissue culture media is the inorganic elements, some sugars, some organic substances like vitamins, amino acids, antibiotics. Antibiotics are very rarely used in the plant tissue culture practices. and some undefined natural growth adjuvants, which are called as the organic supplements, like coconut water is there, yeast extract is there, banana extract is there, potato extract is there. Some other organic supplements are also used in the plant tissue culture at various stages. And then plant growth regulators. These are the major controllers of the plant's tissues in the in vitro conditions. Most probably or universally, auxins, cytokinins, and gibberellins, they are used as the at various stages of the in vitro growth of the plants, especially flowering plants. So these are the composition which is the commonly used for the formulation of the plant tissue culture media. So, dear friends, which are the steps which are involved in the propagation of the plants in the through the plant tissue culture technique? Especially, we can take the example of the micropropagation. If we took the explant from the naturally or the field-grown plants, the first is the establishment of the plant in the culture vessels, then a multiplication of the shoots, then rooting of the in vitro developed shoots, and most important step is, or before transformation of these derived tissue culture derived plants into natural localities, is the to check the genetic assessment or the genetic integrity among the in vitro derived plants. Because here the major motto or the goal of the plant tissue culture technique is the production of true type plants, which means the tissue culture raised plant should be or exactly looks like to their mother or donor plants. And how we are going to assess this genetic integrity? Most of the times they are morphologically or phenotypically alike. They are looks like same. But if we need to check either these are what we can say that similar or dissimilar at the genetic level, there are different types of the molecular markers which we can employ to assess the genetic integrity or genetic uniformity across the tissue culture raised plants. And then accordingly we can make the plan of transformation of this well acclimatized and hardened plants into the natural localities. Nowadays, as I told, nanomaterial is considered under the gold biotechnology type for the area. Nanomaterials are nowadays used in the plant tissue culture for various ways, because in plant tissue culture we need to optimize the surface sterilization conditions while establishing the cultures, and at that time there are chances of the fungal as well as the bacterial contaminations. We are using surface disinfectants like mercury chloride, sodium hypochlorite, chromine water and bromine water. However, apart from this, some of the plant species, they are facing some infections during the in vitro establishment due to the systemic infection. To overcome such systemic infection, one can use the silver oxide, then zinc, titanium, aluminum, gold, cobalt, different types of nanomaterials during the media preparation. So it will help to reduce the contamination in the plants. Here you can see that in the initial stage of this plant establishment, there is the infection. But if we add the nanomaterials before, while preparation of the plant tissue culture media, day by day or slowly, whatever contamination is there, that contamination is reduced or finished within few days, and then the plant grows normally in the plant tissue culture. These are some of the advancement in the plant tissue culture nowadays by using the nanomaterials in the culture media. So dear friends, if we have to, what is our purpose? Depending on that, we are using the culture vessels for the plant tissue culture. We need to develop a protocol that one can use the culture tubes or the test tubes for the culturing of these plants. However, if we need a plants in many numbers or the large numbers, or any industry require the plant biomass in large number, then the culture vessel requirement is different. One can culture it in culture bottles. Then nowadays, the particular industry, they design their own bioreactors for getting the large biomass for particular use. If we need to provide, or if we need to provide the large number of plantlets for conservation purpose, we can propagate such kinds of the plants in the large culture bottles or the plastic plots. That's why we will get the required number of the plantlets within the stipulated time period. So which are the ways through which we can conserve the plants using the tissue culture techniques? As we saw in the micropropagation techniques, one can use the nodal buds or the stem as the explants, and we can produce the number of shoots and then rooting and then hardening. However, some of the plants, they are producing some specialized structures like tubers, combs, rhizomes, and bulbs. So, using the plant tissue culture technique, we can produce such kinds of tubers, combs, rhizomes, as well as the bulbs. And once we produce in the plant tissue culture technique, one can directly transfer these rhizomes, combs, or the tubers to these natural localities. So this is the one more advantage of the plant tissue culture technique. As I told, any part or any explant one can use as the in the plant tissue culture technique for the production of new plants. So dear friends, this is one of the other way one can use in the conservation of the plants, especially this in vitro flowering and fruiting. Most of the times, natural flowering in the plants is not possible. As most of us are aware that the flower induction is not a simple mechanism. It's a very complex mechanism, and which is governed by the several factors: growth regulators, carbon sources, then environmental conditions. But some of the plant species, they are not flowering in the natural conditions. So one can use the plant tissue culture technique to initiate or to induce the flower in some of these places, and they can transfer the flowered plant to the outdoor conditions, and then there will be the pollination mechanism, flowering, fruiting, and other things. This, especially this type of tool, which is used in the plant breeding programs. Somatic embryogenesis is also one of the viable route. As most of the participants, they are aware that what is the somatic embryogenesis. But few of the students are there. I will just highlight what is the somatic embryogenesis. Somatic means the vegetative. As we are aware that when zygote forms, when two reproductive organs comes together, that is the zygotic embryo production. But while using any vegetative part, we are going to produce the embryo. That is the process of the somatic embryogenesis in the plant tissue culture. And there are different stages while maturation. First is the globular stage, then heart-shaped, then torpedo-shaped, then cotyledonary stage. And then one can able to get the what we can germinated plants or the germinated embryos into whole plants. So this is also one of the important method which one can use for the conservation of the plantlets. There are several number of plant species which are propagated through somatic embryogenesis process. Then synthetic seed production is also one of the viable way. Especially this synthetic seed production using sodium alginates and other gelling agents. This method is particularly used for the long-term conservation under controlled conditions. Long-term conservation means one can conserve the shoot tips, nodes, embryos, or seeds in the gelling agents or the sodium alginates, and they can be stored in the cryoprotectants or the especially in the liquid nitrogen at -196 degrees Celsius for several months and several years. And when it is required, one can take it from that cryoprotectants and again it can be regrown. So this is the one of the important aspect of the plant tissue culture and the cryopreservation in combination for the conservation of the plants. For several years, we can keep these synthetic seeds into the cryoprotectants. So dear friends, one more additional feature of the plant tissue culture is the production of secondary metabolites. We are producing the plants in large numbers by using the plant tissue culture. That is one of our major objective for the conservation. But along with this, one can produce the plantlets or the elite clones of the particular species who is having more accumulation of the bioactive drugs, and especially in the medicinal plants. So one of the other importance of the plant tissue culture is the, as I told, secondary metabolite production. But not only secondary metabolite production, how one can boost or one can increase the accumulation or the production of the secondary metabolites by supplying the elicitors. Elicitors are nothing but some of the chemical sources, chemicals extracted either from the biotic and abiotic sources, and that can be added in the plant tissue culture medium. And due to this elicitors triggers the production of particular secondary metabolites or the bioactive drugs in the plant tissue culture, especially callus culture and cell suspension culture. These are the ways where one can trigger the secondary metabolite production using the elicitors and precursors. So dear friends, this is the all about plant tissue culture technique, which is used in various ways to conserve the plants in situ, ex situ, metabolite engineering. Then one can use this technique for the haploid production, as I told. That is the, if we culture the anthers, then we will get the haploid plants. The hairy root culture is there. These are the different advantages of the plant cell and tissue culture. So now we'll turn towards the which are the success stories for the usage of the biotechnology, especially plant tissue culture, for the conservation of the plants. Especially this group of the plants. This is the popularly called as the lantern flowers. That is the Ceropegias. Scientific name is the Ceropegia. And these are the plant genus which comes under the family Asclepiaceae. Now according to the APG3, it comes under the Apocynaceae. This is the plant group which is having over 200 flowering plant species across the globe. But most of the plant species, they are falls in either one category of the IUCN red plant list, which means rare, endangered, critically endangered, vulnerable, extinct, because these flowering plant group have the pollination barrier. Very few plants there produces the follicles or the fruits. If fruits are there, most of the times the seeds which are produced in by these flowering plants, they have the germination limitations. So due to this plant group is day by day, the many species have troubling their existence in the wild. So plant tissue culture technique is the only way for conservation of this plant group. Here you can see that we have propagated the several numbers of the plantlets in the plant through the plant tissue culture technique. We have hardened it at our glasshouse, and over 12,000 saplings of this particular plant species, they have transferred to the various localities in the what we can say that in the northern Western Ghats. Because why these plant species are going, as I told, there is a pollination barrier, there is a pollination, the scarcity of pollinators are there. Apart from this, most of the local peoples, they are uses this plant as a leafy vegetables. Most of the cowboys, they are uprooting the tubers of this plant species and they are eating raw. So that's why those species are on the verge of extinction day by day. So such kinds of, we need to identify such kinds of plant groups for the conservation priorities. And most of the funding agencies, like DBT, DST, and MOEF, they are providing funds for such kinds of the restoration or the reintroduction programs. One of the other species, that is the Abutilon ranadi. Very few numbers of the individuals are of this Abutilon ranadi. They are there in the northern Western Ghats. It is the endemic plant of the northern Western Ghats. And only few numbers are there in the wild. And by using the plant tissue culture technique, we have produced over 10,000 saplings of these Abutilon ranadi plants. And now we have already transferred these plantlets into the various localities in the northern Western Ghats. Not only we have transferred such plants into their natural localities, but we have the record, we have monitored the growth and self-perpetuation of these plants in their natural conditions. And we have got over 85% survival of these plant tissue culture raised plantlets into the natural localities. And in this regard, in this lantern flower as well as Abutilon ranadi, we have got the support from the forest officials, and they are monitoring the growth and development of this plant in the natural habitats. So this is the coordinated efforts of the funding agency, researchers, as well as the forest officials for conservation of this rare, endangered, and medicinally important plant species. These plants have been transferred to the various localities in the northern Western Ghats. And now they are in very good condition. They are now self-perpetuated. They are now growing well in the natural conditions. Dear friends, this slide might be might be seen several times as my mentor, Professor Dr. Yasar Yadav sir, shown in various presentations of him. But again, I have taken this slide from his presentation because I am the part of this restoration program. As I told, Dipcadi, Concanens, and Camptorrhiza indica, these both are the cormatous plants, and these are only grown at the several patches in the district Ratnagiri in the northern Maharashtra. And at that place nowadays, the construction work is going on and several buildings are coming up at that place. So instantly we can't imply the biotechnological tool for conservation of this plant, but a group of researchers from the Shivaji University, Kolhapur, I was the part of that team, we were uprooted that plants from that particular locality in the from the district Ratnagiri of northern Western Maharashtra. So we were uprooted these plants, and again we were transplanted these plant species of similar environment, identifying the various localities from the nearby areas in the northern Western Ghats. We have transplanted these plant species in the various areas, and that's why this Camptorrhiza indica species is now in existence. Otherwise, their plant species might be extinct from this Earth planet, because the construction work is going on at that particular place, and nowadays we will not be able to identify, or we will not be able to collect the plant species from the Ratnagiri. But nowadays it is growing in the adjoining areas of the Ratnagiri in very well conditions. So these are some of the instant and viable routes to conserve the plant species. So dear friends, apart from this, several molecular marker technologies which are now in use for the policy making. These are not directly involved, but policy making regarding the conservation of the particular plant species. There are different types of the markers like biochemical marker, DNA-based markers, as well as the phytochemical markers. One can assess the diversity among the plant species based on this. Apart from the morphological or the phenotypic markers, these are the markers which are commonly used in the genetic diversity assessment across the plants. So biochemical markers one can use for the taxonomic identification, genetic diversity assessment, then evolutionary and ecological studies, then identification of the cultivars and the lines of the same species. So this can be identified on the basis of the biochemical, especially one can use the isozymes. As most of you are aware that the isozymes are nothing but the multiple forms of the particular enzyme. So one can use these isozymes or the proteins. Then you might be heard about the omics technology, which is currently used more powerfully in the diversity assessment, that is proteomics, genomics, then metabolomics. This omics technology is particularly used in the current scenario for the assessment, genetic diversity assessment of this various flowering plant species. Then one can use the phytochemical markers. For example, genus gingiber is there. One can aware about what is the exact chemical composition or which are the major chemical compounds are there in the rhizomes of that particular species. Especially gingerols and chagols are there in the ginger rhizomes. So one can assess different cultivars or different species of the gingers or the whole ginger genus. One can assess for that particular chemical, and on that basis one can consider as there is that how much difference or the which cultivar is close or the nearest to which cultivar. So that phytochemical markers can be used in the assessment of the genetic diversity. And other forms of the most important molecular markers are the DNA based markers. There are different numbers of molecular markers, especially random RAPD is there, SCAR is there, SSR, ISSR, RFLP, RAPD, SNP. There are different kinds of markers. They are used. All these are the PCR based markers. So these PCR based markers are used to assess the genetic diversity. What is the importance to assess the genetic diversity? Once you know which is this cultivar or which is this species which is allied to between them. So if we develop a propagation technique for a particular species, one can use that same propagation technique or the plant tissue culture technique to its allied species. So this is the importance of these DNA marker based techniques in the conservation routes. So as I told, all these are the PCR based markers. One can assess the diversity among them, which are the close relatives. So minimum efforts is required to those closely allied plant species, but one can assess the genetic diversity based on the molecular marker tools. These molecular markers are also helpful for assessing the genetic integrity of tissue culture raised plants. As I told, one can randomly choose the plants, extract the DNA and assess it for its diversity. So based on that, we will also able to know what are the differences between if any plant is missing either any bands. So one can consider it as the soma clonal variation, and that soma clonal variant has the important role in the breeding programs, because that soma clone can be superior to the other plants if that particular especially in the agricultural traits. So that soma clonal variation can be assessed by using these molecular markers. So these are some of the important molecular markers which are generally used in the genetic diversity assessment, which leads into the formulation of the conservation policies for the rare, endangered and medicinal plants. So dear friends, at the end I am going to tell you that is there are several types of genetic manipulation for the yield and traits, especially agrobacterium tumefaciens mediated transformation is there, RNAi technology is there, molecular farming technology is there, which is especially used for the production of the secondary metabolites by using the plant tissue culture technique. So these are some of the important aspects of the biotechnology which can be used for the conservation of biodiversity, not only conservation, so improvement of the qualitative traits among the what we can say that our cultivated crops as well as using the wild relatives of the crop plants by the using gene transfer technology. So dear friends, that's it from my side, as I was more focused on the plant tissue culture because this technique is most popularly used worldwide for the conservation of the plant species. For improvement of the agricultural traits, one can use the gene transfer technology, but plant tissue culture technique is most popularly used in the conservation of the plant species. So that's it from my side. If anyone have any queries or doubt, I'll be happy to answer you. Thank you.
Thank you, sir. You have taken very good account of what is biodiversity, the biodiversity at the heritage site, that is Karst Plateau. What are different tools and techniques to restore the biodiversity, and the actual practical work that you have contributed in restoration of number of species. Thank you very much. Now I request all the participants, if you have any doubt, the consortium is open for your discussion. अगर किसी को कुछ पूछना है इस बारे में तो आपके लिए ये पोडियम ओपन है.
One can ask directly, or you can put it your query in the chat box.
Yes, you can write your doubts in chat box also, or you can unmute yourself and please ask your doubts. Jayanti Gaikwad message is there. Excellent presentation. And it gives us valuable information. By the way, sir, I have one doubt.
Yeah, sure, sir. Please.
Number of species like as Ceropegia, it has pollination barriers.
Right.
Now, although we restore those species by using biotechnology, how they will survive in nature?
Right. One problem is with the scarcity of pollinators, and other is that plant species is having tubers, and that tuber have only one node, and from one tuber one can produce only one plant. And it is not like as our potato. From single potato one can produce several numbers of the plants. There are several numbers of birds are there on the potato. But here, this is the one limitation. Second limitation, as I told you, is the scarcity of pollinators. But what is the way? If we are remaining these plants as it is on its own in the nature, then in the coming future it will be extinct from the its origin, that is from the natural localities. But plant tissue culture is the only way. We are producing these plants and again transplanted into the its natural localities. But nowadays we are monitoring these plant species. They are producing the flowers, they are producing the follicles, that is the fruit which is produced by the Ceropegias, and again their life cycle is going on. But we don't know how that seeds are either viable or not viable, how they are going to produce next generations. We have to assess that populations for the generation after generations.
Yeah, thank you, sir. There is one question from one participant. Sir, please, can you share this PPT?
Sure. I will send it to you, sir. Then you can circulate among the participants.
Yeah, okay.
Thank
Ashwini
you,
madam, you
Thank
can
you,
send
sir.
your email so that you will get the copy of PPT.
Okay, sir.
Okay, thank you very much, sir. And now I request the head of the department, Dr. S.Y. Jadhav, sir, for the vote of thanks.
Thank you all the delegates and participants and dignitaries on this platform. So our institute have organized a one-day national webinar on the eve of World Nature Conservation Day on the role of biotechnology in biodiversity conservation, which has been discussed very well. So,
Yes, बोला. Back camera.
we have discussed well in brief account of this role of biotechnology in conservation of different plants, and I would like to thank to Honorable MLA Vinay Kore, President of this Education Society, whose constant support and encouragement inspired us to conduct this webinar. I also offer my sincere thanks to Honorable Professor Dr. Vasanti Rasam Madam, the Administrative Officer of our Education Society, for her constant support, due to which it has become possible to conduct this webinar. I am also thankful to Dr. Jaikumar Chavan, who have delivered his speech on the issue of conservation of biodiversity by using modern tools of biotechnology. Dear friends, I must mention here that our botany department is running certificate course in biotechnology since 2005. Up till now, more than 1,000 students have taken benefit of it, and many students have acquired jobs in various sections of biotechnology. This lecture will be helpful to all the students and staff of science in general and biotechnology students in particular. I offer my warm regards to Dr. Chavan sir for his inspiring speech. He also given very good future points for the research. It is my duty to offer my sincere thanks to in-charge principal Professor Ph.D. sir for his constant motivation to conduct this webinar. I must offer my sincere thanks to Dr. Khoth, IQAC coordinator and a colleague of my department. His technical skills have enabled us to conduct this webinar. Last but not the least, I offer my warm thanks to Dr. Tarannu Mulla for her introduction of Dr. Jaikumar Chavan in lucid language. Lastly, I offer my sincere thanks to all you participants. Up and always, there are about 168 participants who have participated from different states like Annamala University, Goa, Rajasthan, New Delhi, UP, Gujarat, and other states of the India. Without your support, it was quite impossible to achieve this endeavor. Thank you, one in all. We are sending the link for feedback. Please give the feedback so that you can get the certificate. So thank you very much, Dr. Khoth, Dr. Principal Chikurudekar sir, and all other dignitaries. Thank you very much.
Thank you, sir. Thanks to all the participants. Thank you, Dr. Chavan sir. And with the permission of the president of this webinar, I declare the conclude of this webinar. Thank you.